I am Dr. Silvan, Assistant Professor, PG and Research Department of Biochemistry, St.Joseph College of Arts and Science, Autonomous, Cuddalore. Now I am going to present the topic DNA damage and repair mechanism. DNA is easily damaged under normal physiological conditions. The return of damaged DNA to its normal sequence and structure is called repair. Many different kinds of physical and chemical agents damage the DNA. Some of these are endogenous agents and exogenous agents. Some examples for agents that damage DNA are radiations, highly reactive, oxygen, radicals, ionizing radiation, and ultraviolet rays. The another examples for the agents that cause the DNA damage are chemicals in the environment like aromatic hydrocarbons, including some found in cigarette smoke, and then plant and microbial products. One of the examples are aflatoxins produced in moldy peanuts. Another, the chemicals used in chemotherapy, especially chemotherapy of cancers. Next, the type of DNA damage. The type of DNA damage are the single base alteration, two base alteration, chain breaks, and cross linkage. This table shows the type of damage and the examples. The biochemical mechanism for repair of thymine dimers. The four major pathways dealing with thymine dimer in DNA. The first one examples are light dependent repair and light independent pathways. The another three distinct mechanisms are excision repair, recombination repair, and SYS repair. The first one is photoreactivation. The photoreactivation is a enzyme cleavage of thymine dimer. It is activated by visible light approximately the 300 to 600 nanometer. The enzyme is called PR enzyme or photolyase. These enzymes are almost present in all animals from bacteria to animals. The light energy to cleave the C double bond C bond of the cyclobutyl rings. The first enzyme bind specifically to the thymine dimer. If light absorbed, the thymine dimer was monomerized. Finally, the photolyase dissociates from the thymine dimer. Now the normal pair of thymine is formed. The photoreactivation was processed by the three mechanisms. One is initiation, repairing, and release. In initiation, photolyase enzyme bind to the DNA and creates enzyme DNA complex. And then repairing mechanism, once the absorption of visible light and activation of the enzyme, and then the thymine dimer is monomerized. Finally, after the repairing, the release of enzyme. The another DNA repair mechanism is excision repair. The excision repair essentially consists of removal of a segment of DNA containing the damaged portion of one strand of DNA and new synthesis of the removed segment of the DNA strand using the undamaged strand as a template. The first step is known as incision step. It involves recognition of the damaged segment by an endonuclease. The enzyme cleaves the phosphodiester bond of the sugar phosphate backbone at the 5' end, about 8 nucleotides ahead of the damaged site, producing the 3' hydroxyl group was free. The next step is known as excision step. It involves a cut at a site 4 to 5 nucleotides downstream from the damaged site. This reaction was catalyzed by a 5' to 3' endonuclease. Thereby, a segment of DNA, including the damaged site, is removed. In the last step is known as repair step. The DNA polymerase 1 synthesizes a new stretch of DNA strand starting from the 3' hydroxyl end using the intact complementary strand as template. Finally, the newly synthesized strand is joined with the 5' end by DNA ligase to complete the repair. The another variation of excision repair is catalyzed by the enzyme glycosylase. The glycosylase, which cleaves the N-glycosidic bond between a thymine of a thymine dimer to the sugar phosphate backbone of the DNA strand. At the next step, the phosphodiester bond is cleared by the endonuclease activity of the same enzyme, which recognizes a blank deoxyribose without a base attached to it. In the following step, DNA polymerase 1 initiates DNA synthesis at the free 3' hydroxyl end, displacing the thymine dimer along with a few more adjacent nucleotides. Next, the SOS repair. SOS repair refers to the DNA repair system, which uses Rec-A regulatory protein to inhibit the repressor activity and activate the SOS inducer genes to recover the DNA damage. The SOS stands for Save Our Soul. The SOS response system refers to the mechanism in which an organism initiates the production of activator protein Rec-A, which results in the dissociation of Lex-A repressor and activates the SOS inducer proteins. The SOS repair system is an error-prone mechanism as it works without a DNA template. The SOS system is composed of the following components. The first one, regulator protein. It is encoded by the Rec-A gene, whose function is to activate the repressed SOS system by inhibiting the binding of Lex-A to the SOS operator. The next one, repressor protein. It is encoded by the Lex-A gene, which causes the inactivation of inducer proteins. The repressor binds to the operator and causes inactivation or repression of the SOS system. The another one is inducer proteins. They are encoded by SOS box genes that can activate the inducer proteins relative to the type of DNA damage. In case of excessive DNA damage, stress conditions, a cell responds by activating signal or Rec-A protein. A Rec-A protein specifically binds to the single-stranded DNA. On binding with the single-stranded DNA fragments, Rec-A forms a filament-like structure around the DNA. Then a Lex-A repressor comes in contact with the nucleoprotein filament assembled by the Rec-A protein. When Rec-A interacts with the repressor protein, it converts into Rec-A protease. The formation of Rec-A protease causes autocatalytic proteolysis of Lex-A repressor protein. Thus, a Lex-A protein could not bind with the SYS operator. The inactivation of Lex-A protein activates the inducer proteins that repair the DNA damage but alters the DNA sequence. After DNA repair, the Rec-A protein loses its efficiency to cause proteolysis, and the Lex-A protein will again bind to the SYS operator or switch off the SYS system.